Welcome to the lecture on visual pathways. In this lecture, we will talk in detail about the visual pathway of the neuron, starting from the retina in the front and going all the way back to the occipital cortex of the brain. At the end of the session, you should be able to describe the topographical anatomy of the retina. You should be able to identify the 10 different histological layers of the retina, and then you should be able to describe the anatomy of the visual pathways, as I mentioned, starting from the retina in the front, going all the way back to the occipital cortex of the brain. So what is the retina? Shown over here is a horizontal slice that has been taken through the eyeball. You can see the cornea and the lens in the front and the optic nerve at the back end over here. And where is the retina? Well, the retina can be seen as this innermost light-sensitive tissue layer of the eye, which is color-coded in brown or orange over here. As the light enters into the eye, the optics of the eye, including the cornea and the lens, they create a well-focused two-dimensional image of the visual world on the retina. The job of the retina is then to translate those tons of photons into electrical neural impulses. These neural impulses are then sent back to the brain through the optic nerve all the way back to the occipital cortex, where the visual perception begins to happen. So what are cellular layers inside the retina? Well, the retina comprises of various neural cells. These are organized to form different layers inside the retina. The cells in various layers are then interconnected by synaptic connections. Among the various cells inside the retina, first we've got the photoreceptor cells. These are the primary light-sensing cells inside the retina. These are the first-order neural cells inside the visual pathway, and they can be categorized either as rods or as cones. The rods are extremely sensitive to low-intensity light and hence work well in dim light conditions, and they therefore provide a black and white vision. Cones, on the other hand, function at high-intensity light levels and hence function well in well-lit conditions. These are responsible for the perception of the color and subserve high visual acuity tasks such as reading, for example. Down below over here, you can actually see a cut section which has been taken through the retina. You can see the 10 different layers of the retina shown over here. They've been stained with hematoxylin and eosin, so the neuronal cell bodies are going to appear kind of bluish or purplish in color because of being stained by the hematoxylin stain, whereas the axons of the neurons, they're going to be stained pinkish in color because of the eosin stain. You can see the 10 different layers of the retina over here, and you can see the outermost layer which is shown over here. That is the pigmented epithelial layer, and outside that you can see the choroid, so that will be the outer side of the eyeball. On the inside over here you can see the vitreous body, so that's the inner side of the eyeball. The rods and cones can be seen over here, so this would be our photoreceptor layer inside the retina. Next, we've got the bipolar cells. These are the second order neuronal cells along the course of the visual pathway. These are grouped together to form the outer nuclear cell layer, which is shown over here, or the inner nuclear cell layer. And see, the nuclear cell layer are going to be stained with hematoxylin, so they appear blue in color. The axons which come out from the nuclei give rise to a meshwork of neurons over here, forming the outer plexiform layer from the axons which come off from the outer nuclear layer, or the inner plexiform layer from the axons which come off from the inner nuclear cells. So these are the bipolar cells, and they are the second order neuronal cells inside the retina. Then we've got the third order neuronal cells, which are the ganglionic cells, forming the ganglionic cell layer, which is shown over here. Once again stained with hematoxylin, and these are the nuclear cells, and hence they're going to appear blue in color. The nerve fibers of the ganglionic cells then are going to project out of the retina, forming the nerve fiber layer, and these nerves are then going to project out of the retina, running inside the optic nerve and projecting onto various structures, including the lateral geniculate body, superior colliculus, and the pretectal nucleus. And we'll talk about these structures in detail either as part of the visual pathway or as part of the visual light reflexes in some subsequent lectures. So how does the retina look like in real life when we do a fundoscopy? Now, this is an image of the fundoscopic view of the human retina of the left eye. This region in the center of the retina is called as the macula or the macula lutea. This is linked with the central vision, and there are very few rods but mostly cones present over here. So the number of the cones basically increase, the density of the cones basically increases as we move more towards the center of the retina compared to the peripheral part of the retina. In the peripheral part, we'll see more rods over there. So even though the number of the rods are much higher compared to the cones, the number of the cones per unit area basically increases as we move towards the center. And that region over here in the center is the macula or the macula lutea. Then this little pit or depression, which we can see at the center of the macula or the macula lutea, that is known as the fovea or the fovea centralis. And here we can see the fovea contains only cones. There are no rods at all present over here. So this region subserves the photopic vision. It is linked with the central vision, and this is the region of highest visual acuity. Now, if we resort to this section over here, see, this is a section once again of the retina. We can see the different layers of the retina, which we saw previously as well. You can see the different layers stained by the hematoxylin and the eosin stain. You can see the vitreous body over here. So that's the inner side of the eyeball, and the choroid with those vessels inside it can be seen on the outer side of the retina. And what you can see over here in the region which is the fovea or the fovea centralis, you can see that the different layers of the retina have kind of been displaced laterally, and as a result the photoreceptor layer, which is the layer of rods and cones, that's been totally exposed to the light directly. And this pit or depression which is created over here, that is the hallmark of the fovea or the fovea centralis. Now, coming back to this fundoscopic view of the retina, you can see that medial to the macula or medial to the fovea centralis over here, this region is known as the optic disc. It is about 3 to 3.5 millimeter nasal to the macula lutea, and it contains axons from the ganglionic cells and the retinal vessels. You can see the retinal artery splitting up into different branches over here. So as there are nerve fiber layers over here popping out from the ganglionic cells, and there are loads of blood vessels over here, they kind of displace the photoreceptor cells towards the outer side, and consequently in this region we do not have any photoreceptors inside the retina, and therefore this area creates a kind of a blind spot in the visual field. However, luckily we don't perceive that, as our brain predicts and fills in that blind space inside the visual field based on the data from the surrounding regions inside the visual field. Now, you might be wondering by now that how come the fovea or the fovea centralis be the area of highest visual acuity in the retina? Let's try to answer that question by once again looking at this section through the retina, which shows the 10 different layers of the retina, stained again by hematoxylin and the eosin stain. Once again, the choroid can be seen on the outer side, and the vitreous body can be seen on the inner side over here. You can see the 10 different layers over here. You can see that the different layers are kind of covering over the photoreceptor layer, so the light cannot actually directly hit upon the photoreceptor cells. But if we look over here, so this would be a section through one of the peripheral regions of the retina. But if we go towards the central part of the retina where the fovea or the fovea centralis is present, what you can see is that the layers, as I mentioned previously as well, the layers have been displaced laterally, and so the photoreceptor cells are in direct contact with the light. That is one of the reasons why the fovea becomes the area of highest visual acuity. Then as we go towards the center of the retina, the number of the cones per unit area starts increasing. They are much more in number compared to the rods in the macula, and by the time we reach the centermost part of the retina, which is the fovea or the fovea centralis, there are no rods whatsoever, and the cones are maximal in terms of their density, and that contributes to the highest visual acuity of this region as well. Now, before we embark upon talking about the visual pathways, there are two important principles which we need to understand. We can take the help of this cartoonic illustration over here. This is a cartoonic drawing of the visual field, and you can see that we can divide the visual field over here into two halves, the right visual field on the right side and the left visual field over here. Similarly, this is a cartoonic illustration of the retina. We can see the retina of the right eye over here, the retina of the left eye over here, and we can divide the retina into two halves as well. We've got a temporal retina on the outer side and the nasal retina on the medial side towards the side of the nose. Similarly, we can see the nasal retina on the left eye over here, and we can see the temporal retina of the left eye over here. Now, the first important concept which you have to understand is that the visual field is flipped over when it gets represented on the retina, meaning thereby that the right visual field, which you can see over here, that's going to be projected onto the left side of our retina. So therefore, we can see that the right visual field is projecting onto the left part of the right retina, which is the nasal retina, and it's being projected onto the temporal half of the left retina. Similarly, if you look at the left visual field, you can see it's being projected onto the right side of the retina, that is onto the temporal retina of the right eye and the nasal retina of the left eye. The flip is not just in a horizontal plane, it's going to be happening in a vertical plane as well, meaning thereby that the objects in the upper half of the visual field, they're going to be projected onto the lower half of the retina, while objects in the lower half of the visual field, they'll be projected onto the upper half of the retina. So for example, if you're looking at me, my image is going to be flipped over on your retina, right? My head is going to be projected onto the lower retina of yours, and my legs are going to be projected onto the upper retina of yours. Later on, the information gets projected onto the visual cortex. So the information from the upper retina goes to an area of the visual cortex which is known as the cuneus, and the visual information from the lower retina goes to the area of visual cortex which is known as the lingula, and we'll talk about those in a bit. So for now, I want you to hold on to this piece of information, and it's going to be really instrumental in helping you to understand the lesions across the visual pathways and helping you to understand the visual deficits which occur as a result of lesions across the visual pathways, which we will talk about in the subsequent lecture. Here again, once again, you can see the same principle being explained over here. The only difference is what I've done over here is that I've actually split up the visual field. This visual field, I've just split it up into separate visual fields for the right and the left eye, but the same principle is being explained over here as well. The right visual field, you can see, is being projected onto the left retina, on the nasal half of the right retina, and the temporal half onto the temporal half of the left retina, and vice versa. Now, the second most important principle which you have to know is that the circular retina is represented on the cortex in such a way that the central part of the retina, the macula or the macula lutea, containing the fovea and the fovea centralis, that is projected onto the posterior part of the occipital cortex, right? So this region of this is a cartoonic illustration which is actually showing you the medial surface of the occipital cortex. You can see the calcarine sulcus over here separating the occipital cortex into a gyrus above, which is known as the cuneus, and a gyrus below, which is known as the lingula. The primary visual cortex is situated around the calcarine sulcus, and the central part of the retina, the macula, which is the area for the highest visual acuity and is concerned with color vision because it has the highest density of cones in it, that is represented onto the posterior part of the occipital cortex, whereas the paramacular area or the peripheral part of the retina, that contains predominantly cones for peripheral vision and lots of rods for the dark vision. That area is actually projected onto the occipital cortex in the primary visual area anterior to the macular area. So the retina is represented in such a way that the central part is represented on the cortex at the back end over here, whereas the peripheral part is represented more anteriorly. Now, this is really important to know. Once again, this information is going to help you understand the lesions in the visual pathway, which we will talk about in the subsequent lecture. But for now, What you have to know is that this area at the back where the macula is represented, that has kind of a dual blood supply to it. So we've got two cerebral arteries, the posterior cerebral artery and the middle cerebral artery, which both supply this little region over here, or which could potentially supply this little region over here. If we look at the medial side of the occipital cortex, we can see this area is primarily being supplied by an artery which is known as the posterior cerebral artery. But then there's another artery called the middle cerebral artery, which is predominantly supplying the superolateral surface of the brain, but sometimes it can potentially come onto the medial side as well and supply this little region where the macula is represented. And this is a kind of a protective mechanism which is being offered by the nature, because we are really much more dependent on our central vision for our survival compared to the peripheral vision. So we can actually still live with the loss of peripheral vision, but we need the central vision for high visual acuity tasks such as reading, for example. And so the dual supply in this region basically spares the macula if in case there's a stroke involving the posterior cerebral artery, causing a deficit of vision in most of our visual field, but that still spares the central vision because of the fact that the middle cerebral artery from the outside comes for the rescue. So what is the course of the visual pathway neurons? Let's begin by orientating this pro-section and this illustration over here. So what you're looking over here is basically the base of the brain. You can see the olfactory lobes in the front. These are actually the orbital surfaces of the olfactory lobe. You can see the olfactory bulb and olfactory sulci over here as well at the front. We are interested in this nerve over here, which is the second cranial nerve. That is the optic nerve. You can see the optic nerve kind of combining with each other, and their fibers are going to cross over here. This region is known as the optic chiasma, and we'll talk about that in a bit more detail. And then from the optic chiasma, you can see the optic tracts coming out, and then these then go back over here to an area which is known as the lateral geniculate body, which is part of the thalamus. And then the fibers, they spread out to form optic radiations, and they go all the way back to the occipital cortex. Much of the temporal lobe and some of the occipital lobe over here has been resected, so you can actually see the optic radiations passing through those lobes. And we'll talk about those parts in a bit more detail, but now we're just trying to orientate the pro-section. This is an illustration of the same visual pathway over here. Now let's talk about the different parts of the visual pathway in detail one by one. So the visual pathway, if you look over here, basically starts from the retina. You can see the retina of the right eye over here and the retina of the left eye over here. As we discussed previously as well, when we were talking about some of the important principles which we have to understand in context of the visual field and the visual pathway, we said that the retina could be divided into an outer temporal half and an inner nasal half. Same thing is happening here as well. This is the nasal half of the left retina. This is the temporal outer half of the left retina. Now what you can see here is that the optic nerve fibers, they pop out from the base, from the back of the retina, and they run inside the cranial nerve, which is the second cranial nerve, that is known as the optic nerve. Each optic nerve is going to be having fibers coming from the temporal retina and fibers coming from the nasal retina. Now these optic nerve fibers are going to pop out through the optic canal into the cranial cavity. So the eyeballs are present inside the orbital cavity. The optic nerves are going to be originating from the back of the retina inside the orbital cavity, but then they pass through the orbital cavity through a canal which is called optic canal, and after passing through it, the other end of the canal is going to open into the cranial cavity. So the optic nerves now enter into the cranial cavity, and that's the part which you can see over here. These optic nerves can be seen crossing over here in an area which is known as the optic chiasma. We already identified that crossing part over here, the optic chiasma at the base of the brain in this pro-section over here. Now from the optic chiasma, we can see the optic nerves continuing as the optic tracts now. So these are the same neurons, but they're just given different names in different places. We call them optic nerves over here. Then they cross over here at a region called optic chiasma, but they're still the same nerve fibers. And then after crossing, these regions are known as the optic tracts. The optic tracts then extend backwards and they actually go and synapse over here in this region, which is known as the lateral geniculate body. Lateral geniculate body is one of the nucleus of the thalamus. And remember, just as a basic rule, that whenever we talk about any sensory pathway, every sensory information has to relay inside the thalamus. There is one exception though, the olfactory sensation does not relay inside the thalamus, but apart from that, every sensory information has to relay in the thalamus before it goes to the higher center, such as the cerebral cortex. And therefore the visual pathway, the visual information, is no such exception either. And so the part of the thalamus where the visual pathway is going to relay the visual information inside the thalamus is known as the lateral geniculate body. Now before we actually go further from the lateral geniculate body all the way back to the occipital cortex, let's just understand some of the crossing of the fibers which is happening over here. So what you can see here is that the optic nerve, which contains the temporal and the nasal fibers, these fibers, when they actually reach in a region called the optic chiasma, the fibers actually cross over. And this is a very common thing which happens inside the neuronal tracts in the brain. The crossing or the decussation happens at different levels inside the central nervous system for different sensory and motor pathways. Now what you can see here is that the temporal fibers, they do not cross at the optic chiasma, but the nasal fibers are the only ones which cross over. So in other words, if we take up the optic tracts, for example, let's say if we take the right optic tract, for example, we can see that the right optic tract will receive the temporal fibers from the same side or from the ipsilateral side. However, it will be receiving the nasal retinal fibers from the opposite side or from the contralateral side, in this case from the left side. Similarly, if we take up the left optic tract, we will see it will be receiving the temporal fibers from the same ipsilateral side, that is the left side, but the left optic tract would be receiving the nasal retinal fibers coming from the right side, its contralateral right side. Now, we also know that the visual field flips over when it gets projected onto the retina. That is the right visual field that projects onto the left retinal halves, and the left visual field projects onto the right retinal halves. Therefore, if we take up the right optic tract, for example, we will see that it receives the nasal retinal fibers from the contralateral side and the temporal retinal fibers from the ipsilateral retina. And since the temporal retina here and the nasal retina over here gets the left visual field projected onto them, therefore the right optic tract receives information about the left visual field. And if we apply the same logic over here on the left side, we will see that the left optic tract will be receiving information about the right visual field. And you can do the math and you can check it if that's true or not. Another important thing which I need to mention over here before we actually extend backwards from the lateral geniculate body is that, see, have a look at this important relation over here. We can see the optic chiasma over here. If we look at the base of the brain, the optic chiasma is very nicely visible over here. Right behind the optic chiasma, you can see this region over here. That's the base of the brain. This is an elevated region in the base of the brain, which is known as tuber cinereum. And from here, the stalk of the pituitary gland is going to pop out, and the pituitary gland is going to be hanging down below, a little bit anterior but inferior to the optic chiasma, sitting inside a bony space inside the cranial cavity, which is known as the sella turcica. And so in other words, what I'm trying to say is that the pituitary gland is going to be the antero-inferior relation of the optic chiasma. And if people who get some enlargement of the pituitary gland, let's say a pituitary adenoma, which is a common reason of the enlargement of the pituitary, that adenoma, if it is a macroadenoma and big enough to compress the optic chiasma over here, people usually would present with visual deficits. The usual visual deficit is the bitemporal hemianopia, and we'll talk about that in the subsequent lecture when we go over to the visual pathway lesions. But for now, just remember that the visual impairment is an important clinical presentation of a macroadenoma inside the pituitary gland, and that's something which is really important to know from a clinical standpoint. Now, till here, we are actually talking about the ganglionic nerve cells, the ganglionic nerve fibers, which were traveling in the optic nerve, in the optic chiasma, and in the optic tract, reaching up to the lateral geniculate body. Now they're going to synapse over here, and the new neurons which start over from the lateral geniculate body as part of the visual pathway, they are going to project out and radiate to form optic radiations prior to reaching the occipital cortex. The superior optic radiations, they travel through the parietal lobe and reach the occipital cortex in the primary visual area, while the inferior optic radiations, they pass through the temporal lobe and reach the primary visual area inside the occipital cortex. Now, in order to understand that, let's have a look at this prosection over here. As a first step, let's try to orientate this prosection. So what you're looking over here is a medial view of the cerebral hemisphere. This is a mid-sagittal section which has been taken anteroposteriorly through the brain, and we can see the medial view of the left cerebral hemisphere over here. The occipital lobe is clearly shown over here, which is bounded by the parieto-occipital sulcus. In fact, the parieto-occipital sulcus is separating the occipital lobe from the parietal lobe in the front. Within the occipital lobe, we can see a sulcus over here that is known as the calcarine sulcus, which is going to split up the occipital lobe into two gyri. There is a cuneate gyrus or cuneus at the top, and there is a lingual gyrus or lingula down below. The primary visual area lies inside the occipital cortex on either side of the calcarine sulcus. So basically inside the upper lip and inside the lower lip of the calcarine sulcus. So partly inside the cuneus and partly inside the lingula. The superior optic radiations, they take information from the superior retina, and therefore, in other words, from the inferior visual field, and they project onto the cuneus part of the primary visual area inside the calcarine sulcus, whereas the inferior optic radiations, they bring information from the inferior retina, or in other words, information about the superior visual field, and these then project, after passing through the temporal lobe, they project onto the lingula or the inferior lip of the calcarine sulcus. So that is how the visual information is brought from the retina all the way back into the occipital cortex in the primary visual area around the calcarine sulcus. Now remember that the upper visual field is projected down below into the lingula, and the lower visual field is projected up into the cuneus. The crossing is not just in a vertical plane, it's in a horizontal plane as well. So the right visual field is going to project onto the left occipital cortex, and the left visual field is going to be projected onto the right occipital cortex. In other words, we can actually divide the visual field into 4 quadrants. So we can see, for example, if we take the example of the cuneus on the left side, the cuneus on the left side would be receiving visual information from the right visual field. But the flipping of the visual information of the visual field is in a vertical axis as well. So the cuneus on the left side is not just receiving visual information from the right visual field, but it is actually receiving visual information from the upper quadrant of the right visual field. Similarly, if we take the example of the lingula on the left side over here, we can see that the visual information on the left side is going to be projected from the right visual field. But since the visual field is going to be flipped over in a vertical axis as well, therefore the lingula is only going to receive the visual information from the right visual field only from its upper half. And we are going to revise these concepts one more time when we talk about the lesions of the visual pathway in much more detail in a subsequent lecture. Thank you very much for listening. Hope you benefited from the lecture. If yes, then do please like the video. In the next video, we will talk about the lesions of the visual pathways.